Greetings. Welcome to BrainGage channel. You know, this talk is about a little bit of a deeper dive into the brain gauge measures. And to do that, we really are going to have to we're going to have to start looking at the harder science, the basic science that we spent decades doing, and those are what the measures are actually based on. So, what are building blocks of information processing? I mean, just as a reminder for people who don't know, things like processing speed, focus, lateral emission, plasticity, and timing, those are all things that we measure, that we can measure with a brain gauge. And then, things like language and learning, those depend on those building blocks. Executive functions depend on all the building blocks below them. Cognitive processing depends on all the things below them. So it's really important, to look at, these different building blocks. And if there's anything wrong with these, stuff up here isn't going to work. And, different people are crazy in different ways. And, but you could probably trace it back to something down here. You can always trace it back to some problem with connectivity. But let's take a look at how to study the brain. And what we did was we always deliver a sinusoidal stimulus. That's what the brain gauge does. It delivers a nice, gentle mechanical stimulus. It's very high fidelity. And this is something that you can do with the sense of touch. And you can't do it with the others. But anyway, and then we stick electrodes in the brain and we would get recordings. And those recordings would look something like this. You have a stimulus, and this is a response to that stimulus. So what do you do with this, and how can you study anything? Well, one way you can do it is clean it up a little bit and do the math. Everybody wants to do the math and convert these neural recordings to what we call our spike trains. And it turns out that all neurons or the nerves or the cells in the brain, they all fire. When they fire, they have a specific size or shape. They're very unique. And like, for example, what we do is we just want to say, when does this particular neuron fire? In this case, we've got 3 different size units on this picture here. You see these dotted lines are threshold lines. And this one has gone above this line, but it hasn't gone above that one. So there's a spike here. This one has crossed this threshold, but it did not go above this line. So it gets a mark here, separate from this one. And here's another one. It's above this line, but not there. So what you do is you start to see these patterns of spike trains, and you look at how neurons fit in with the overall network. I'm just going to give you some really simple stuff. This is like first day of class kind of stuff. But anyway, when we look at the periphery, and we look at how well the periphery is, like if you were to stick a nerve, if you were to stick an electrode in your wrist, and you basically want to record, basically you're recording these same spikes in the periphery as the same time the stimulus is going on. And you see an extremely regular pattern, right? Well, it turns out that regularity is not in the cortex. There's a lot more variability in the cortex. Information is actually in the variability. But anyway, so how do you do something with what do you do with this? Well, one thing you can do is you can convert this to the circle histograms. And the way you do that is remember, sine wave it looks like this, and it can be converted to here. So one spike on a sine, if the spike occurs during that sine wave, during the sinusoid, you'll get one spike here. Now in the periphery, it's very regular. So all these things are lining up in about the same place. In the cortex, in this particular case, it's a little fuzzier, but it's still contained within one quadrant. Whoops. Still contained within one quadrant. And, basically, though, you can see the difference between the periphery and cortex, but this is just a mathematical representation of the difference between the two patterns. So what do we see? So one thing to remember is that the brain, what we see is when we have a stimulus here, this is control stimulus. This is the same stimulus, but with an insult provided, and then another control stimulus. So what happens? First, you see a fairly focused radial histogram here. You see a messy histogram here, and still a messy histogram here. And this radial histogram comes from these trials, these first few trials. Then when the insult is provided, we are no longer following that pattern. The entrainment is no longer attaching as well to that sinusoid. The brain is no longer working as well. And then even after the insult is removed, that pattern remains. Now, many of the measures of the brain gauge depend on how well these things fit, how well they fit to that pattern. And the reason you can do these tests, or if you can do them, if you can, a lot of these tests, particularly things like amplitude discrimination, if you can do the accuracy test fairly well, then you're probably looking like this. If you get a significant insult to your brain, now this, and that's not an insult in the same part of the brain where we're recording from, or where you're actually performing these tactile functions or mechanical functions. It's actually in a different part of the brain. So the insult somewhere else, all right, but it has an impact on the part that's doing this mechanical assessment, the brain gauge scores. And that's something that a lot of people have trouble comprehending. I won't go into who that is. They're very high level people. They've been given jobs and where they get paid a lot to make bad decisions. But anyway, it's all the parts of the brain are connected. That's usually pretty easy for most people to contemplate and figure out. So if you need to know any more questions, you need to know anything else, go to the website, corticalmetrics.com. If you have a question for me, or you want to see a video made or anything, just email me at thebraingauge@gmail.com, or free consults on my calendar. So if you have any questions about the brain gauge, I'm happy to talk. So that's it, and have a good holiday if you're watching this before the holidays.